Redox reactions are chemical processes where electrons are transferred between chemical properties. If we split redox itself, we get reduction and oxidation. Reduction is where a property gains electrons, while oxidation is where it loses electrons. Reductants induce other substances to reduce, while oxidants induce other subjects to oxidize. Reduction is also characterized by the gain of oxygen and loss of hydrogen atoms, and oxidation is regarded as the gain of oxygen and loss of hydrogen atoms. These characteristics become critical in photosynthesis, a redox reaction in its own right, by reducing carbon dioxide into glucose and oxidizing water into oxygen gas. Oxygen drives cellular respiration for organisms such as ourselves. And glucose is utilized by plants, firstly as an immediate source of energy, secondly to produce energy-dense storage, and thirdly to produce cellulose, critical in strengthening cell walls. These three operations are simply means of ensuring that oxygen production is sustained, so that our cells can decompose food into energy. Unfortunately, photosynthesis isn't this simple, but fortunately for you guys, I'm here to explain. The light absorbed by photosynthetic pigments excites present electrons into a higher energy state, converting light energy into chemical potential energy. An electron pair is then transported through the stroma to one of two photosystems, simply units within photosynthesis. The P680 special pair of the second photosystem, pigments best at absorbing 680 nanometer light, is where the electron's journey commences, before being transported through the electron transport chain, a progression of molecules that undergo reduction and then oxidation as electrons pass through them. Now, P68 is extremely electronegative. So upon losing its electron pair to the electron transport chain, it becomes positively charged with a proclivity to not remain so. In order to restore a natural charge, electrons will be extracted from surrounding water molecules to produce oxygen gas and hydronium ions, the latter being expelled from the electron transport chain and into a matrix of intermembrane space. This is due to its aversion to water throughout the plant. We would then call them hydrophobic. So instead, these protons are transported through this intermembrane space through hydrophilic tunnels of the ATP synthase, an enzyme synthesizing ADP and phosphate into ATP, a DNA building block essential in photosynthesis. For now, think of ADP as a worn-out version of ATP, with one less energy-supplying phosphate group. The moving protons will generate what is known as a proton gradient, rotating the ATP synthase like air to a windmill, catalyzing the recharging of ADP into ATP. Then these excited electrons are transported to P700, best at absorbing 700 nanometer light, of the first photosystem to then reduce NADP positive into NADPH. From here we enter the Calvin cycle, a progression of molecules converting CO2 into glucose. At the cycle's initiation, the 5-carbon bound compound RUBP synthesizes with carbon dioxide to then break down into 2 of the 3 carbon bound compounds 3-PGA. Then each 3-PGA is donated a phosphate group from the newly recharged ATP molecules, producing ADT as a byproduct, which is returned back to the ATP synthase for recharging. 3-PGA is now doubly phosphorylated, but not for too long, since NADPH, a strong reductant, donates an electron pair to substitute this phosphate group out from this rapidly changing molecule. Now we have NADPH, phosphate, and a newly formed G3P molecule, the actual resource required for producing glucose, or some of it. In order for this cycle to self-perpetuate, most G3P molecules are recycled back into producing the RUBP acceptor, commencing this entire cycle. See, for every 3 CO2 molecules expended, 6 G3Ps are produced, with only one of these spared to produce glucose. See, over their entire journey, electrons jump from molecule to molecule, which, from our knowledge of oxidation and reduction, is essentially just a complex string of redox reaction. As molecules accept these electrons, they undergo reduction, but upon donating them to the next molecule, they undergo oxidation. So on the surface, we do have the reduction of CO2 and oxidation of water into glucose and oxygen, but this is only because CO2 receives its electrons from NADPH to produce 3-PGA, and that water loses its electrons to the extremely electronegative P680. So we know that G3P and glucose, in respect to all other life, are produced as an energy source to sustain the primary function of oxygen production, and as a material enhancing their own physical strength to prolong their lifespan. It is purely by this oxygen that we can decompose food into caloric energy, applicable within all domains of life. And while we've only ever discussed in reference to plants, plankton, accounting for 72% of the world's oxygen production, also undergoes photosynthesis. Moreover, these life forms are a predominant solution to global warming. Essentially, mass carbon dioxide production exacerbates global warming due to the greenhouse effect, resulting in increased spans of drought loss of critical species, and an exacerbation of the factors leading to poverty and displacement. So by reducing atmospheric CO2 emissions by its consumption in photosynthesis, plankton and plants foremostly intensify our contemporary environment and economy's vibrancy, but then also sustain this stable state into the future. Thus, determining the best conditions for photosynthesis is crucial to ensuring an optimum future. And to aid this mission, I've chosen to evaluate the influence of red, green, and purple light on each plant's proficiency to photosynthesize, especially within photosystem 2, by measuring the amount of oxygen produced. To do so, I've fashioned closed systems for each identical aquatic plant with tubing connecting them to this water displacement system. Essentially, as oxygen passes through these tubes and up into the bottle, its minimal density will cause it to push the bottled water down into this reservoir and reside at its peak. While color light exposure is irregular for plants, promising data from any one of these plants could provide sufficient evidence to normalize plant colored light exposure. If integrated, plant color light exposure would maximize production of any essential function reliant upon photosynthesis, such as agricultural industries. Unfortunately, no data arose due to the unprecedented volume of oxygen required to generate enough pressure capable of forcing gas through this tubing and into the bottle. And no plant this small is going to do that in a week. Yet as a duty to our future, and in order for me to get more marks, we can still identify the light best fit for photosynthesis optimality. From an ill-educated perspective, we would assume that whatever light excites the electrons greatest allows photosynthesis to occur rapidly, and thus more proficiently. I mean, electrons do lose energy when they're traveling through electron transport chains, and would thus slow down, suggesting that more energized electrons would complete this photosynthetic process quicker. And under these presumptions, purple light would transfer more energy to these electrons due to its substantial frequency. However, the special pairs P680 and P700 will, as we know, absorb light to a greater extent if it is 680 or 700 nanometers in wavelength. Purple light lies between 380 and 400 nanometers and will thus be poorly absorbed by any of these special pairs, reducing the rate at which electrons are supplied sufficient energy to actually instigate photosynthesis, let alone making the whole process less expedient. Rather, shades of red lie between 650 and 700 nanometers, indicating that since light energy is better converted to electrical potential energy at this wavelength band, the red exposed plant would have produced the most oxygen. This is in consequence to the increased rate electron pairs are excited, and the rate these electrons travel through each photosystem to convert these reactants into these products. So if you're ever in a predicament over the best source of light to optimize plants' photosynthesis, choose red. This guy could not have thought of a worse assignment. I swear. Who are you? It's time, my child, for a change of new. From blue to black, I'll change your view. A blanket of coal I will provide, but the color to follow, you'll decide. Choose red. Choose red. Red.